Hey everyone, so today I'm going to go over the stuff that we talked about last class with like the Christmas tree model of how we look at transcription and translation occurring in bacteria versus in eukaryotes. So I'm going to focus on the bacterial model here. So this is an image that we saw in class of DNA and mRNA and ribosomes sort of all in the same image. So I guess I'll just start off with ribosomes are generally represented, since they're so large, they're represented by like the big black spots that you'll see. And we know that ribosomes read mRNA. So whatever the ribosomes are attached to, you know is mRNA. So like in this case, we see that they're attached to this, this strand here. So this we would label as our mRNA. So I think that's like the first place to start if you're given an image like this, is to just, if there are ribosomes, they're pretty obvious to tell like where they are, and those are what are going to be attached to your mRNA. And then we know that mRNA is generated from DNA. So whatever your mRNA is like sort of coming off of would be your DNA. So this would be the DNA. And then I think the main point where people were getting confused is how to determine the directionality of everything. So I'll draw sort of like 3 time steps. So I'll draw like time step 1, 2, and 3. So I'll draw the DNA in blue. So I'm just going to draw one strand for now. So I'm just going to draw it like as the template strand, just so that we get a sense of what's going on. Okay, so let's say that RNA polymerase A binds and starts moving along the mRNA, reading the template strand and generating mRNA. And then let's say that like 10 seconds later in time step 2, RNA polymerase B binds, and then A is like somewhere over here, and they're both still moving to the right. So A has read this whole section of DNA, right? And so now it's turned that into mRNA. So out of the polymerase, there'll be like a little strand of mRNA that we've made that corresponds to this length of DNA. But now B has, it started at the same start point as A, right? Because polymerase has to bind to, in prokaryotes, it has to bind to sigma factor, and then it'll start moving towards towards reading the gene. So then at time step 3, let's say that polymerase C binds, B has moved a little ways, and A has also now moved past like where it was in time step 2. So everyone is still moving to the right. And now A has read this much more than it did up here. So it'll be much longer now, the mRNA, because it's read much more DNA, versus B has now only read this much. So it'll be kind of like that big, approximately. And C hasn't read anything because it's just starting at the same place that A and B started at. So as you can see, the longer the mRNA strand is, the earlier that polymerase had started compared to the other ones on the strand. So if we look up here in our example, this mRNA is much longer than these ones over here. So we know that this one had to have started earlier and these ones started later. So like maybe this one started at like time equals 0 seconds, and then this one was like, I don't know, like 0.5 microseconds later. Like I don't really know what the time magnitude is or whatever, but just to get a rough, like just a sense of this, the longer mRNA we know was started but making by a polymerase that started a lot earlier than the ones on this side, because on the left side of the DNA they're so much shorter, so the polymerase actually hasn't read much of the template DNA. So because of that, since we know that the way that transcription works, the polymerase will read along the DNA in the 3' to 5' direction, and it starts at the 3' end because it reads 3' to 5'. So then we can say that this end is the 3' end, and this end is the 5' end, right? Because we're starting over here, the strands are smaller because the polymerase hasn't actually moved as much along the DNA. It hasn't read all of the template DNA. So we know that the starting point has to be on the left, and that's the 3' end, because we know that transcription begins at the 3' end, because that's how polymerase reads. And then from there, we can actually determine the mRNA directionality, because we know that the template strand and the mRNA are complementary to each other. And so because they're complementary, we know that they have to be anti-parallel. So what I mean by that, if I redraw this, I'm just going to draw A on its own somewhere. So let's say this was like start, and it's moved along there. So if I cut this, and then I line up this strand with the DNA, so I line it up, this is 3', 5', oops. Then I know that this end has to be 5' and this end has to be 3', right? Because these have to line up. These bases have to be complementary. So I know that the end sticking out is the 5' end because it was complementary to the 3' end, right? It was the first base that was transcribed was corresponding to this 3' end. So I know that the 5' end has to be sticking out. So all of these are the 5' ends. Don't actually know exactly where the ends are, but just to get a rough idea. And then if we look further, we can actually zoom in, if it'll let me. Yeah, okay, it's kind of blurry, but it actually isn't shown in this drawing, but those ribosomes, right, they're starting at the 5' end and they're moving down the mRNA strand, because translation occurs from 5' to 3', right? So a ribosome would bind and then it would move down the mRNA and make protein, right? It's putting amino acids together. So in this case, every ribosome, the ribosomes down here had started way up here. So they've had way more time to translate than the ones that are at the 5' end. So the proteins sticking out of those ribosomes will be much longer than these up here. And then we know that proteins are made in the N to C terminus direction, right? If I have a generic amino acid in the R group, and so let's say this is R1, this is where my peptide bond will form. So I'm always adding to this end, and so this is my N terminus, and eventually, right, I'll add more and more. The final sort of amino acid that's added, so I'm just going to call it R infinity, but it's not actually an infinite amount. This would be our COOH, right? CO, and then this is COOH. And so this is our C terminus. And so you can see that this is going to be the first thing that sort of sticks out of the ribosomes. So hopefully that helps and gives like a general idea. Here, I'll just label that for clarity. So that's the N terminus that's sticking out of the ribosomes. Yeah, so hopefully that's helpful in sort of just illustrating all the moving parts of what's kind of going on here. But I think the most important thing to remember is the fact that because this is prokaryotes, transcription and translation can happen at the same time, versus in a eukaryotic cell, we wouldn't see this because the mRNA has to be transported outside of the nucleus and into the cytoplasm where translation would actually occur.